All right, everybody, thank you so much for attending. It's really good to see so many familiar faces. So many of you guys have been with the company since we started this crowdfunding, since day one, back when we were a preclinical research company before we graduated to this clinical stage. So without further ado, I'm Joey Bose. I'm your president and CEO coming from live from West Palm Beach, Florida, and I'm here to talk about Cytonics. Cytonics is a privately held biotech company headquartered in Jupiter, Florida, with the primary directive of developing the first and only true disease-modifying therapy for osteoarthritis. And as Nietzsche said, he who has a why can endure any how. And our why is simple. We are addressing big pharma's failures that have led to over 500 million people suffering from osteoarthritis, debilitating pain, and completely annihilated quality of life. And we simply find this unacceptable. This clear-cut why is what drives our how, which is to address big pharma's failures, their myopic approach to drug development, and simultaneously reclaim biotech innovation by ripping it from the grasp of Silicon Valley, big pharma, and Wall Street, and place it directly in your hands, the patients, the physicians, the people that need it most. Osteoarthritis is an epidemic. We often think of it as a disease of aging. And while it is chronic in nature, it affects everybody, even young people, athletes. Over 32 million Americans currently suffer from OA, and over 500 million people worldwide. And you might not believe it, but that represents a $1 trillion burden to the global healthcare system. Clearly, this is an unmet clinical need that demands a solution. But a true therapy, a real solution, requires you to dive deep into the root cause of the disease, the molecular pathogenesis, which we've identified. It's a class of enzymes called proteases, and these proteases are catabolic. They chew up other proteins, including the proteins that compose cartilage. So a real disease-modifying therapy, meaning a therapy that's not merely palliative, has to address these protease enzymes that become hyperactive in arthritic joints. And unfortunately, none of the currently available treatments actually do this. They're all palliative. They mask joint pain and inflammation temporarily. NSAIDs, hyaluronic acid, corticosteroids. You've heard of all of these things. Unfortunately, they don't bring any sort of long-term, durable efficacy to the patient suffering from osteoarthritis. And in the case of corticosteroids, they're actually deleterious, more damaging than good over the long term. A steroid shot is meant for halftime at the football game. You got to get back on the field. It's not meant for chronic use. Chronic use will actually destroy the cartilage even further. So it's simply unacceptable that there is no durable disease-modifying therapy for osteoarthritis until now. Enter Cyte-108. This is our lead drug candidate that we believe is positioned to be the first and only true disease-modifying therapy for osteoarthritis. And it is predicated on a naturally occurring molecule called alpha-2 macroglobulin. It's a mouthful. We call it A2M for short. And this A2M is a naturally occurring protein that's found in the bloodstream. And you guessed it, it's a protease inhibitor. And it's capable of inhibiting all of those protease enzymes that are responsible for osteoarthritis, therefore targeting OA at the root molecular source. Now, our research and recently verified by an independent academic group also shows that alpha-2 macroglobulin has the potential to induce cartilage repair on the molecular level. This is huge because now it's a brake on the cartilage degradation and the gas on its synthesis. So we theorized if we could deliver high concentrations of an alpha-2 macroglobulin variant that's been engineered for superior function in inhibiting those proteases, that we could develop a solution that's actually durable, targeting the root molecular pathophysiology of the disease, capable of being manufactured in a laboratory and distributed at global scale, something that can get to all of the people worldwide that need it. Cyte-108 is an engineered variant of the naturally occurring A2M molecule. And what we did is we made careful genetic mutations to a very specific region of the protein that binds the proteases. The Cyte-108 can then be injected directly into the joint. This targeted delivery is huge. It's not systemic, which limits its ability to circulate and potentially accumulate in organs. So what we've developed here is a super A2M that's with targeted delivery that's able to be manufactured at scale and delivered globally. Now, our research didn't start with Cyte-108. It actually started by figuring out how to harness the power of the naturally occurring A2M molecule. And that's what led to the genesis of our first therapy, the autologous protease inhibitor concentrate system. Another mouthful, we call it APIC. Every word in that acronym means something. Autologous coming from self. Protease inhibitor, that's the A2M. Concentrate, it is a selectively enriched A2M formulation pulled from your blood using a proprietary technology, a filtration system that we developed. Now, the APIC therapy has treated over 10,000 patients worldwide using the naturally occurring A2M that's purified from their blood. It's a miracle how quickly the A2M formulation, this APIC therapy, is able to instantaneously relieve the perceived pain and inflammation while also offering a durable effect. The average number of APIC injections per year for a patient is something around 2 to 3, with some patients only needing a yearly injection and some patients only needing a single injection. As you can see, there's quite a bit of patient-to-patient variability, and that's one of the limitations of the APIC system in using the naturally occurring A2M that's found in the blood, right? We're going to have different A2M levels depending on all sorts of biological factors from age, sex, general health. One of the other limitations is that the APIC system is a medical device, and it requires time and equipment and training to be able to administer. So we theorized, what if we don't have to actually rely on the natural A2M? What if we could build something better? And that's what led to Cyte-108, the next generation A2M therapy. Cyte-108 addresses every single one of those limitations of APIC by creating a super A2M, genetically engineering the natural A2M gene sequence, so you can have precisely dosed, highly concentrated, improved A2M function that's able to be manufactured at scale and has a high potential for insurance coverage because it's actually going through the FDA's biopharmaceutical pathway, phase 1, phase 2, phase 3 clinical trials. These are randomized control trials, which will definitively, within a statistical doubt, be able to prove its efficacy. And that's what insurance payers want to see. And obviously, as you know, we know in our healthcare system, getting insurance reimbursement is paramount to being able to get people treated. Simply put, Cyte-108 is nature's blueprint perfected. We took the natural A2M gene sequence and we improved it using a technology called recombinant protein engineering. So Cyte-108 is the natural evolution of the therapy. And this might remind you of something else, which is insulin. Prior to 1982, when Genentech and Eli Lilly created their recombinant insulin, diabetics were treated with the insulin purified from the pancreases of animals. So we go from naturally occurring harvested molecule to recombinant, a variant that's been engineered for specific and improved function. Now, before going into humans, we had to experiment in preclinical studies. And our preclinical studies showed that Cyte-108 was exceptionally well tolerated, as evidenced by an absence of abnormal organ pathology, nor a deleterious immune response. Basically means the drug doesn't accumulate in other organs, heart, spleen, liver, and cause some sort of damaging effect, nor does it induce an immune response that leads to patient sickness. This safety information served as a very important predicate for moving into clinical trials. And it was buttressed and buoyed by the preclinical efficacy experiments that we conducted, looking on the molecular level of what Cyte-108 is actually doing in the joint. And what we're able to see was that it reduced cartilage damage by almost 60% while restoring the health of the cartilage secreting chondrocytes by almost 50%. Now, its efficacy doesn't stop at cartilage. We were able to see positive effects on the subchondral bone, which is the bone on either side of your joint, could be knee, doesn't matter, elbow, whatever. That subchondral bone, when the cartilage gets destroyed, starts going like this. And anytime you have this sort of traumatic, chronic grinding, you're going to get sclerosis of that tissue to protect it. So that sclerosis is not a good thing. We're able to reduce the amount of subchondral sclerosis by almost 80%, while also reducing the joint membrane inflammation by approximately 60%. This information in the preclinical served as massive, important information to be able to go into phase 1 clinical trials, both from a medical and scientific perspective, as well as a regulatory perspective. And we were able to do so. Starting in June 2024, we launched our first in human phase 1 clinical trial in Australia. The last patient was monitored in March of 2025, marking the end of the dosing and patient follow-up stage of the phase 1 clinical trial. Now we are cleaning the data, meaning putting the data in a database in a form that a statistician can go and easily analyze, and an independent medical writer can draft a clinical study report. And we expect to have that clinical study report here in the third quarter of 2025. What I can tell you is there have been no reported drug-related adverse events, meaning that the drug appears to be very well tolerated when injected into the knee. Our phase 1 clinical trial looked at the safety and efficacy of CYT108 in primary osteoarthritis of the knee. And that is the first indication that we're going to go for, but it's not the last. It is not limited to the knee. A2M, as we've seen with the APIC system, has efficacy in any joint that contains articular cartilage. Like I said, fingers, toes, wrists, knees, elbows, even the spine. So you can see from the perspective of an insurance payer, this has a potential massive impact on the bottom line. Why? Because it can delay or even obviate the need for total joint replacements, which contribute to about $27 billion annually in the United States. A total knee replacement is on average $33,000. This type of evidence in randomized human trials, it's what's going to lead to the future potential, the economic viability of CYT108 from the eyes in the perspective of insurance payers. That is absolutely huge. But first, we have to de-risk the development through the FDA's regulatory framework, which has 3 independent, discrete phases. Phase 1. The main goal of a phase 1 study is to de-risk the safety. We have to make sure before we start injecting this drug in hundreds and thousands of patients that it's well tolerated. Number 1, the FDA's number 1 concern for everything is safety. Phase 2, now you are looking for efficacy as a primary endpoint. And you don't want to just see trends in efficacy, you need to see statistical significance, meaning beyond a statistical doubt, we're actually able to make claims that the drug is effective. Then phase 3. Now you are repeating your phase 2 and phase 1 experiments in thousands of patients and selecting an optimum dose, which will appear on the drug label and it's what physicians are going to prescribe to their patients. This 3-part system, this 3-phase system, is what comprises the FDA's regulatory framework and is what's needed to get a biologic license application, a BLA. When a BLA is submitted to the FDA and approved, that's when the drug can be commercialized. That's not Cytonics' strategy. We do not intend to go all the way through phase 3 clinical trials at this time. Our goal is to de-risk the development of CYT108 through phase 2, producing a convincing efficacy data set, and then either IPOing the company so that our shareholders have a liquidity event, or possibly licensing the development rights to a larger biotech that has more resources and the know-how to be able to take this into larger trials, because all this costs money. So the question is, why not do it ourselves? It's simply a matter of risk and what's in the shareholders' best interest. So it's very possible that we IPO the company, raise $100 million, and run the phase 3 ourselves, but it's not the only option. And that's very unique, right? Because having this optionality of when we decide to give our shareholders an exit event is very important. And it gives us flexibility, and it gives us the opportunity to, in an incremental way, do what's in the best interest of the shareholders from a financial perspective. So multiple opportunities and a very clearly delineated pathway to those exit events. Our next milestone is going to be a Type C meeting with the FDA in which we go over all of the data collected to date, both preclinical and what's in our phase 1 clinical data set. Once we have that Type C meeting, we are going to file an investigational new drug application, which is going to allow us to come back to the United States and conduct those phase 2 clinical trials. We expect that IND to be submitted in the first quarter of 2026 and the first patient to be dosed in our phase 2 shortly thereafter, which will put us back in front of the FDA for an end of phase 2 meeting sometime in the latter half of 2027. And that's when the exit opportunities, those conversations really start in earnest. Do we IPO? Do we try and sell the company? Or do we enter into some sort of co-development licensing agreement with a larger biotech company? And this is something that is a really good problem to have to solve, and it goes to the board to vote so that the shareholders' best interest is always kept front of mind. Now, when it comes to trying to determine what is the real value of the company, you have to look at the total addressable market and what percentage Cytonics drug, CYT108, could potentially take. Now, it's very difficult to try and estimate the market size for a treatment who has no competitors. There is no disease-modifying therapy for osteoarthritis out there. So how are we going to know how big the market is and what percentage we can take? So we very cleverly looked at the sales of rheumatoid arthritis therapies, a class of drug called TNF-alpha inhibitors that are disease-modifying for RA, which is the sister arthritis to osteoarthritis. Taken together, the annual sales at their peak were about $43 billion of these TNF-alpha inhibitors. The number one best-selling drug of all time until recently was Humira, which generated by itself about $22 billion in annual sales. So we took this $43 billion figure and we multiplied it by the ratio of the prevalence of osteoarthritis to rheumatoid arthritis, which is about 13x, meaning there's 13 times more people that have it. That gives you an over $500 billion market potential and over 500 million people that could benefit from this therapy. Massive market potential, massive total addressable market, and this is what starts the financial wizardry to come up with a valuation for the company. Now, as an investor, regardless of what you're investing in, there are three main questions that come to mind. The first is, why is the company valued at what it is today? What the company is going out, the share price? How is it going to be valued more in the future? And how is Cytonics or the company's management team going to be able to achieve that increase in valuation? Now, for a company like Cytonics and any sort of pre-revenue biotech company whose value is predicated on the research of a drug that's not in the market and is not generating revenues, this can be very difficult. You're not assigning a PE ratio to a publicly traded company and looking at the balance sheet or looking at the income statement and looking at their earnings and trying to estimate, what a good share price would be, what a good valuation would be. Instead, what we have to do is try and impute the valuation from looking at the future potential economic returns of the drug. Now, we can do that by looking at the potential cash flows over the viable economic lifetime as defined by the patents, somewhere between 15 and 20 years, and sum them and adjust them for risk. And that risk comes from two factors. Number one, there's an opportunity cost to everything. So your money could go somewhere else. So you have to account for that. Number two, there's inherent sector-specific risk in running clinical trials. So we need to discount those cash flows to the present, right? $1 today is worth more than $1 tomorrow. So this is called a risk-adjusted discounted cash flow analysis. And that exercise is what's produced the figures on the right-hand side of the slide based on the assumptions on the left-hand side of the slide. So you can see, applying these conservative assumptions, 50% discount rate to accommodate for the risk, a 20% market capture, saying that we're not even going to be able to take the full $500 billion market, and a dosing schedule, a revenue model very similar to APIC, which is about $2,000 per treatment, average $2,000 a year. That's what produces a risk-adjusted discounted cash flow that shows what the valuation could be as it moves lockstep with each stage of clinical trial success. While it moves lockstep, it does not move linearly as you can see, there is an exponential increase in the potential valuation. Now, I should disclaim that all of these figures are derived from a forecast. All models are wrong, but some models are useful. And I find this one particularly useful. So what we're giving you today, the reason I'm sitting in front of you, is to present you with an opportunity to join a grassroots effort to invest like a venture capitalist in a company that has accepted $0 from traditional VC. And in doing so, we've insulated our cap table. We've been able to maintain majority interest. Our founder owns almost 20% of the company still. I'll own, on a fully diluted basis, somewhere between 5 to 10%. Clearly, our incentives are aligned with all of our shareholders. And in doing so, we're going to challenge Big Pharma's stranglehold on innovation, as well as Silicon Valley, VCs, and Wall Street's grasp on the industry. We have over 7,000 retail shareholders, all coming from crowdfunding. I've raised about $25 million to date. We're raising, at this point, we have about $5 million from the ongoing crowdfund, and we'll probably hit somewhere between 6 and 7 million total. Enough that we're going to be able to fund our phase 2 clinical trial by the end of the capital raising that we're going to do over the next 12 months. So what this opportunity is for you to join this populist grassroots effort and invest in a biotech that's disrupting the traditional VC financing model and disrupting the way that drugs are traditionally developed under the umbrella and the stranglehold of Big Pharma. We're a ragtag group. Our founder is a spine surgeon and 5th degree black belt in jiu-jitsu. Real dangerous guy. He does not come from Big Pharma. Stanford trained, amazing spine surgeon, the best on the East Coast, maybe in the entire country. Myself, I come from a biomedical engineering background. I worked on Wall Street. I saw how horrible it was. And that's when I met Dr. Scuderi, the founder. And I joined Cytonics back in 2018. Neither Dr. Scuderi nor myself come from this traditional background. And it's this different perspective that's going to allow us to succeed, where the traditional model has clearly failed. In the appendix of this presentation, which you'll get, I've outlined the 5 biggest Big Pharma failures. Now, me and Dr. Scuderi and our board can't do it ourselves. This last year, I've assembled the osteoarthritis dream team. 6 individuals, all MDs, some of which are MD PhDs, who under their collective belts have 80 osteoarthritis clinical trials. They've been there, they've done that, and they're playing a very important role in the design of our clinical development plan, in the design of these osteoarthritis clinical trials. They've seen why they've failed, and they're going to be able to address those root failures so that we don't have to repeat them again. Our track record is truly astounding for a company of our size and as lean as we are. We already have proof that we can navigate the FDA's very complex regulatory framework with the clearance of our APIC system as a 510(k) medical device, which has treated over 10,000 patients, de-risking the development of Site 108 by proving the efficacy of A2M, the A2M protein, which it's predicated on. We've received almost $2 million in non-dilutive financing from the NIH to support our early research, and we've raised a total of $32 million to date, not including the round that's ongoing. Me, myself, I've raised $25 million from over 7,000 equity crowdfunding shareholders just like you. With our recently completed phase 1 clinical trial, we've proven we can get into human studies and advance further. With our robust intellectual property portfolio, we have the ability to protect the commercial potential of this drug. It's insane, truthfully, what we've been able to do with our current capabilities, and it is a learning process every single step of the way for all of us. But we're getting it done, and we invite you to join us. So with that, I will end the presentation, and let's get into the questions. We got plenty of time. All right, I'm going to go to the Q&A. I know some people have been dropping things into the chat. If you wouldn't mind, drop them into the Q&A just so I don't have to go back and forth. All right, let's see. Thomas, greetings from Silicon Valley. It's good to see you again. So Thomas asks, If time permits, please share your thoughts on Plan B, just in case FDA roadblocks are too steep. Excuse me. Get some water. The name of the game is incremental de-risking. We are not Pfizer. We're not swinging for the fences. What we are doing is designing clinical trials that will incrementally de-risk every single aspect of the development. That includes the trial design, things like the types of patients that are recruited, the demographics, the dosing scheme, the dosing levels, even what you are looking for as an endpoint. So our strategy here isn't to just take the 10 million, 20 million, throw it against the wall, one big trial. Even if we experience a failure, it is going to be informative, and it's going to allow us to run a second or third study. This next study is going to be a phase 1B/2A, meaning we are probably going to conduct an underpowered study, meaning in less patients than needed to get statistical significance, but be able to look at multiple doses over a window, what we think will be a therapeutic window. And those results are what are going to inform a larger phase 2B/3 properly powered, meaning enough patients to get statistical significance, right? Now, in addition to this osteoarthritis program, we are about to make a big announcement where we're expanding the development pipeline to a completely new indication. I'm not going to say what it is right now because I'm waiting for the provisional patent to be granted. If I tell you exactly what we're doing right now, it'll technically be a public disclosure, and I really shouldn't do that on the internet. I expect to have that patent next week. Once the provisional patent is executed, we'll be able to disclose this new disease indication we're going after using Cyte 108 in our plan to do it. So in expanding the development pipeline, another aspect of de-risking. Charlene asks, will the presentation be available afterward? Absolutely. And I'll be sending out a separate email. And the call is recorded. You'll get a recording of that as well. Jasper asks, which is a great question, how are you going to get certification on a true disease-modifying medication? What's the FDA going to consider to be disease-modifying? Is it X-ray, arthroscopic evaluation? This is where we're pioneering. We have to make the case to the FDA that what we're looking at, the effects that we're measuring, actually indicate disease modification. Because let me tell you something. Even if we were to restore the joint back to normal, healthy physiology, like an 18-year-old athlete, if there isn't a clinically meaningful benefit as defined by a patient's improvement in pain and quality of life, none of that matters. So what we first have to establish is that there's a clinically meaningful benefit, pain, essentially, and improved quality of life. There are patient-reported outcomes that capture that constellation of efficacy, and then go after the disease modification. We have a few ideas on looking at certain structural changes using MRI and X-ray that we believe will define disease modification, the restoration of the joint back to a normal, healthy state. Now, those details are going to be discussed in that Type C meeting. So we're not going to proceed to the disease modification portion of the trial until after the pain has been established, number one, and we have the FDA's input so that we can feel confident that if we see the structural changes that we expect to see back to the normal, healthy physiology and anatomy, that we'll be able to get that disease-modifying label, which is the holy grail of the entire sector of regenerative medicine. David asks, what are the big differences between previous A2M and Cyte 108, especially in terms of efficacy versus cost? So the previous A2M, which is our first generation technology, that APIC system, which selectively enriches the A2M that comes from patients' blood, is an autologous therapy, right? It's purifying natural A2M. And so, as you can imagine, there's patient-to-patient variability. It takes time, equipment, training. It's not something that can be easily scalable. But from a competitive pricing standpoint, we expect to be able to create commercially viable Cyte 108 that sells at the same price as the APIC system, which is about $2,000 a treatment. Now, of course, we're going for insurance coverage. So the insurance company will be billed something else. That's a whole commercial analysis that we're in the process of conducting. It's very important to do. And as we move through and we get closer and closer to a potential exit, that commercial analysis, very important. Now, the efficacy, we've already seen that the Cyte-108 is about 3 times more effective as a protease inhibitor compared to the naturally occurring A2M, and we expect to see increased efficacy over naturally occurring A2M in these human clinical trials. What is the effect or impact, if any, of the current tariffs on overseas investors? Well, there are no tariffs on investments, so you don't have to worry about that. The only thing that could possibly be an issue is manufacturing. We have 2 GMP runs, which are the manufacturing runs undergoing right now, and it's certainly not affecting our finances in any meaningful way. Jasper's follow-on question about pain, is that going to be the first indication? Yes. So as mentioned before, this is an incremental de-risking process. The first indication we're going to go for is pain, then disease modification. Mike asks, what additional long-term financing plans are planned after August 2025? Bonds, stocks, and what's your timing? So only equity. We're not issuing any sorts of convertible notes. We plan on completing this financing on August 27th, at which time we are going to focus on publishing that clinical study report, come out soon thereafter. And we'll open up another round. We haven't decided on the timing or the valuation yet, but this current valuation, this is the only time it's going to be at this current share price, which is $3 a share. This raise plus the next raise is going to generate enough capital to be able to get through our phase 2 clinical trial, which we've already priced with a number of contract research organizations to be anywhere from $3 million to $10 million, depending on the scope and where we actually conduct the trial. So we're going to be fully financed through phase 2 by mid next year. Jasper asks, in phase 1, do you collect data on pain control? Absolutely. This is randomized controlled. The placebo is saline. Barry asks, will this treatment help remedy a rotator cuff injury? So I can't say that definitively because we're not actually going after soft tissue modification. However, one of the indications that we're thinking about going after, which again, I'm not going to make a public disclosure until we have the provisional patents, has to do with soft tissue. It's not rotator cuff, has to do with soft tissue, but rotator cuff is also something that conceivably could work. And here's why. Those soft tissue diseases like rotator cuff injuries, like epicondylitis, are mediated by proteases as well. And that's why we think of our Cyte-108 development as actually a platform technology where we can go in and tweak that little bait region that binds the protease for different proteases that are responsible for different diseases. And we're going to prove that when we expand our development pipeline later this year. Jasper asks, time to exit. Phase 2 will likely be our exit, which will be end of 2027, early 2028. But again, I need to qualify it. This is management's forecast, right? And this obviously is a very fluid situation. So I don't want to give guarantees on exit value or timing, but early 2028 is a pretty good stopping point, exit point to keep in mind. David asks, well, if a knee replacement is $33,000, what is the cost of Cyte-108 per dose? We're pegging it right at $2,000. That's going to be a commercially viable figure. James asks, are you also looking at the veterinary, particularly canine use of Cyte-108? Absolutely. And our APIC therapy has already been used in horses. It's working for equine veterinarians, both performance and working horses. We have a licensing agreement with a veterinary distributor who was just bought by a private equity group for APIC in the veterinary market. Dogs are next. And honestly, it could be a much faster way to get to market for Cyte-108. So it's definitely something that we're keeping in our back pocket. Not the primary focus, but if the right partner, if we can find the right partner, pursuing approval through the Center of Veterinary Medicine is a very, very viable path forward. Jasper asks, how long will it actually take to get disease-modifying classification? I estimate the trial will take 2 to 3 years. And that's based on a study of loricivivant, which is a WNT, a Wnt pathway inhibitor. It works, its mechanism of action is completely different than Cyte-108. But they were able to see structural improvements as quantified by changes in cartilage volume and joint space narrowing starting at year 2. and persist and improve out to year 3. So disease modification is a long-term goal. Pain comes first. Jasper asks, do we know the valuation after this round? What will the price per share be in the next round? Haven't locked it in, but as you can imagine, there's a valuation increase predicated on success. Ron asks, how can we enter the trials? So my email address is on the bottom right of this slide. Feel free to email me. Now, obviously, our phase 2 has not been locked down as to where we're going to conduct it, but it's possible, especially if we have a lot of clinical sites in the United States, if there's a clinical site near you. So we'll definitely add you to the list. Greg asks, how and where do I invest? So you can go to our Start Engine page. I'm going to put it here in the chat to place an investment directly. And actually on this next slide, hold on one sec. I lost the presentation somehow. There we go. This next slide, you can see our landing page, this invest.cytonics.com. You can go there and click invest and place your investment. Joe asks, if you've already invested in the first round, why invest in the current round? That is a deeply personal question. And it has to do with your risk tolerance. It has to do with your portfolio and your just overall investing strategy. If you've invested a lot of money in Cytonics and it represents an over allocation in your portfolio, then you should not invest again. You should absolutely do what's best for your portfolio. And, as should be pretty obvious, but I should say, this is a very high risk play. But with high risk comes high reward. So, deeply personal portfolio management, not financial advice, but that's how to think about it. What happens to the company if me or Dr. Scary are hit by a bus? Well, that's not good. We'll have to find a new CEO. I mean, look, you can only have so many depths to management, but let's hope that doesn't happen. It's not going to happen. Very safe. I don't take the bus, and I don't drive like crazy, so we're good. What can expedite the FDA process? Can we move faster? There is an opportunity to follow one of 4 expedited development pathways. One would be a breakthrough designation. And a breakthrough designation would happen if we have convincing human data that the FDA basically says, all right, we need to get this thing to market faster. So what they'll do is they will offer priority reviews, and they will interface with the company on a more frequent basis to expedite the process. But there are 4 development pathways: it's accelerated approval, priority review, breakthrough designation, and accelerated approval. Breakthrough would be the one that we would apply for. Daryl asks, can this type of biotech innovation be useful for all the major sports leagues going forward? I'm going to say yes, because we treated the Eagles defensive line using APIC. We treated Hoist Gracie, who, if you guys are into UFC or MMA, he's sort of the OG. He brought Brazilian Jiu-Jitsu to the map, UFCs 1, 2, and 3. We cured his bicep tendon. You can go on our YouTube and listen to his testimonial. We treated his nephew, Henner. We're tapped into the UFC and mixed martial arts. I started training jiu-jitsu years ago with Dr. Scuderi. He's a dangerous dude, and it's been great to meet a lot of those professional athletes. Living in South Florida, this is the mecca for MMA, and so we have quite a presence, and we're pretty well known in that community. David asks, as an early investor, can you tell me what kind of ROI do you see when Cytonics goes in the stock market or published through acquisition? Our goal is to give the last money in a 5 to 10x, which means for the earlier money, it should be greater. Now, of course, that is a goal. What the actual valuation will be at the time of exit is predicated on Cytonics' intrinsic value, that risk-adjusted DCF, but ultimately, it's the market. Something is only worth what someone else is willing to pay for it. And the market for mergers and acquisitions, same thing as the market for a car, right? It's competitive, it's fluid. And in some cases, you're in a buyer's market. Some cases, you're in a seller's market, just like a house, right? What I can tell you is that over the last 5 years, we've had a pendulum shift from an overly frothy, aggressive IPO and M&A landscape back to a much more tame. But guess what? Pendulums move in 2 ways. I believe we're going to be very well positioned for the pendulum to swing back. Steve asks, how will these be different than stem cell joint treatments? Well, So first of all, stem cells have not been approved except for a single blood disorder from embryonic umbilical stem cells. So they're not actually FDA approved to treat all the things that doctors are injecting them for. Cyte 108 will actually have a label, meaning it's an FDA approval with very specific information that supports it. It's like its efficacy data, its statistical efficacy data. Completely different ballgame. Absolutely different ballgame. Ron asked, does it have any effect on spinal stenosis? That's something we haven't even looked into. Couldn't say. Charlene asks, do you have any concerns about the current, political FDA landscape? What do you think the current administration is going to do? Will they take exception to this therapy? So there's certainly been a lot of turnover. I think that Marty Makary, the new head of the FDA, is extremely competent. And I think he's doing a good job of reorganizing the FDA to make it more efficient. So honestly, I think that things are going to be just fine. We've certainly not had any problems interfacing with the FDA. I don't know of any other biotech CEOs that have been complaining about it. So I don't really see an issue. I think a lot of things, just like everything political, gets really frothy on the news, really evocative. And the truth of the matter is things are usually a little more tame than what the media landscape likes to drum up for views. Sandy asks, is there any potential benefit for knee replacements that have not had a great outcome? Will it be able to help with a joint replacement? One of the things we've been considering is actually using it as a prophylactic. Sometimes you have to have a joint replacement, but that's highly inflammatory, or perhaps an ACL reconstruction is highly inflammatory. If the joint is already open, why not inject it with Cytonics 108? It's not even injecting. Why not just layer it on, right? So that's something we've really thought about because it seems obvious. So that's not the way we're designing our first trial, but it's certainly something that we might pursue because it makes a lot of logical sense. John asks, does it have to be injected by an ortho, rheumatologist, family doctor? Typically by an ortho, but even family doctors are getting really good at these intra-articular injections without even using ultrasound. They do what's called landmark. They'll do landmarks. They'll figure out, all little protuberance here. I'm not a doctor, by the way, so I'm not using the right words. Little protuberance, and then, this is right where we need to inject it. So it's most likely going to be administered by orthos, but I think there are a lot of family doctors now that are perfectly capable of these intra-articular injections without even using ultrasound. David asks, in addition to insurance companies, will you also go for reimbursement from Medicare? Absolutely. CMS is the first one. Absolutely. Ron asks, why did stem cells fail? Stem cells didn't even fail. They never even tried. They didn't even try. It's all marketing. And the reason for that is that it's autologous or allogeneic. So the FDA takes a looser stance on stuff that's pulled from the body and injected back into the body. So they never tried because they didn't need to. It's a multi-billion-dollar industry. You got Joe Rogan on the internet talking about it. Why even worry about FDA qualification if you're selling it? It's kind of crazy. Let's see. Daniel asks, is Cyte 108 designed to permanently regenerate cartilage in all applications? If yes, Cytonics should become a large company. Will selling licenses be the best avenue for margins? So, yes, we would definitely like to position it as something that could regenerate cartilage, but that has to be proven. We've seen molecular indications that cartilage is being synthesized, right? But that would need to be shown on a structural level. We believe that Cytonics could be a massive, massive company. This is basically like developing insulin. The first ones, Eli Lilly and Genentech, to develop insulin. It's such a similar story, right? Now, when it comes to licenses versus having the company acquired, that's something that later, once we actually are in talks with strategic partners or acquirers, that the board will sit and do an analysis and see what's in the shareholders' best interest. And look, at that time, if we have a development pipeline with 2 or 3 other indications, maybe we just IPO. Maybe we license out the osteoarthritis angle and we just keep the licenses for treating other diseases in-house. very possible. And that's a very common model where you'll license out the development rights for a specific disease, and along with those development rights, the commercial rights to manufacture and sell. And you'll start working on other things and just keep building out this cadre of different development projects for different diseases. And in doing so, increase the valuation of the company. Okay, let's see. A lot of good questions here. Brian asks, large market for athletic performance? Definitely. Would there be for young professional athletes who incur sports injuries in addition to OA? So touched on that, absolutely. Think about an ACL reconstruction. You're going to already have to open the joint. Why not put in something that can reduce inflammation and encourage regeneration of cartilage and potentially other soft tissues, sort of in a prophylactic sense? So absolutely. Jeff asks, given the current FDA regime, do you think they're going to promote more fast track approvals for novel therapies? So, RFK came in and said a lot of that. I haven't seen, a lot of follow through, but, we're early into this administration and, this regime of people. So we'll have to see. We'll have to see. James asks, have you had any communications with the biohackers? Because this aligns right with their goal. And the biohackers are, these guys and gals that are trying to extend their lifespan, right? Without necessarily giving a lot of thought to health span. Why would you want to live longer if you're not healthy? So Brian Armstrong comes to mind, right? He's the guy that's like optimized everything down to, the milliliter of water he drinks. And apparently he's restored himself from like a 40-year-old to like an 18-year-old physiologically. He looks like a vampire. I don't know if you ever seen him. His skin's translucent. So maybe physiologically, based on the blood panel, you look healthy, but that guy is crazy. Now, we are tapped into the sports community, the UFC community, and one of the things we want to do is get Joe Rogan a treatment, an APIC treatment, just get him to see, that there's a lot of potential here. Obviously, we're not going to try and get him to, do some sort of deal or anything like that. But the more knowledge there is out there, especially on platforms where a lot of these biohackers watch, would be great. Jasper asks, when you have convertible notes to stocks, we're not selling any convertible notes. It's all priced rounds. It's all common stock. Right now, $3 a share. Anonymous attendee, are treatments on a permanent fix, or do clients have to return yearly for treatment? The average number of treatments, probably 2 to 3 a year for the average patient, some yearly, some every few years, some one or done. This is something, of course, I'm speaking very anecdotally right now. This is something that will have to be proven in the clinical trials and will influence the ultimate drug label that gets on the drug once the FDA approves it, if the FDA approves it. Thomas asks, any plan to extend the deadline for this round? No, this one's closing August 27th. We have a really strong predicate to open another round at a higher valuation. So, that's what we're going to do. Can this drug aid and help speed up other non-invasive regenerative treatments, especially shockwave? We've never experimented with these other types of therapies. I love all that stuff. I do cupping, deep tissue massage. I do shockwave. This is not something that we've paired it with. Those obviously aren't, FDA approved treatments, and, we're following a very specific FDA regulatory pathway here, but who knows? Goes to market, you get a lot of people, doing a lot of things to their bodies. And it's possible that, maybe there's some synergies here, but it's not something that we're looking into right now. Question, if the knees are healed, will that also take care of Baker cysts? So you're right. That's a question for the OA Dream Team. I'm not a doctor. I can't really comment on that. David asks, if there's an acquisition, would the stockholders be able to vote on this for approval? So typically what happens in an acquisition, by our bylaws and just traditionally, the board of directors is going to vote on that in the shareholders' best interest. And there's going to be a damn good reason for it, right? Obviously having individual shareholders vote, on something that important could be an absolute nightmare, which is why the board of directors is so important. And individual shareholders do have the right to nominate board of directors, directors to the board. So as we move forward, we're going to increase the number of directors to get 7, majority independent, and we're going to get some people with a little bit more experience on the licensing, the acquisition side of things, and they're the ones who are going to ensure that whatever deal that we decide to take at that time, if we take a deal at all, is in the shareholders' best economic interest. Kathleen, you said I'm unable to see the questions. Well, I can see your question, so you can just type in this Q&A and I'll get to it. Yeah, Mike makes a comment that, after the ACLs, that's a ACL repair, that's a big market, right? We could stratify patients into they're in surgery or after recovery. Absolutely. And, that patient selection when you go into clinical trials and you do these things is paramount. So a question about Start Engine shares. I initially bought in Start Engine. Should I still manage shares with them? So yes, all of your stock is visible in Start Engine, but more importantly, we keep a log, and a third-party administrator called a transfer agent keeps a log as well. So if you have any questions about your ownership, you can obviously just email me. If and when it comes time to disperse those shares, in the case of an acquisition or an IPO, it's that third-party administrator who's bound by all sorts of SEC, Securities and Exchange Commission regulations that will actually handle that, like sending shares to your personal brokerage account. Okay, and this invest.cytonics.com is just a landing page. When you click invest, it actually goes to the Start Engine portal where you can actually place the investment. It's just an easier URL. And finally, yes, this presentation is recorded. It will be sent back out and along with the deck. So guys, thank you so much. We had some really great questions. As always, it was a pleasure. We came in right under time. So with that, I'll leave you to it. Thank you guys so much.